let's get started. So yeah, kia ora, welcome to our July digital events. On behalf of the team at Neurological Foundation, a huge thank you to you all for coming along and yeah, spending your evening with us, and we hope that you will enjoy the session tonight. Just to get started, as we usually do, I just want to share this short whakatauki with you. It says, e hara tākou toa, e te toa, takitahi, engari he toa, takitini. And that translates to, my strength is not as an individual, but as a collective. And we think that that, yeah, is a nice tie to what our researchers do and what we do as an organisation, with the help of our community and our wonderful donors. So thank you. Shortly, I will be handing over to Dr. Sarah Schonberger to introduce our guest tonight. But first of all, I'm just going to go through some housekeeping with you. So as you can see, your camera and your microphone have automatically been turned off. You'll be unable to turn these on, and this means we cannot see or hear you. Instead, we invite you to use the Zoom chat and Q&A functions if you'd like to communicate with us during the event tonight. You'll find the chat and Q&A function at the bottom of your screen. To use the chat, just click on the icon that says chat. That will pop up a window, and you'll see that it's set that you can only speak to the host and panelists. So no other attendees will be able to see what you write in this box. This is the box that you use if you have technical support questions, and I will do my best to help you with those. If you have any questions you'd like to ask, please use the Q&A function. Again, the Q&A function can be found at the bottom of the screen. You click on the Q&A to pop up a window, and this is where you can put your questions. The questions here are visible by all attendees, and we'll be able to see who has submitted the question. However, if you do wish to remain anonymous, please just tick the Send Anonymously box at the bottom before submitting your question. Once a question is submitted, the presenter can opt to answer it live so everyone can hear the answer, or we may type a response. Because we have a limited time frame tonight, we may not be able to answer all questions. And also, please keep the questions related to the topic presented. Our speaker will not be able to answer any personal medical related questions that you may have. At the conclusion of the session tonight, we'll end the Zoom for everybody. And those of you who have attended before know that this happens quite abruptly, so I'm sorry for that. But the recording will be available on our website at a later date if you wish to revisit it. So now I'd like to hand over to our Head of Research, Dr. Sarah Schonberger, who will introduce our speaker for the evening.
Thanks very much, Amy. Kia ora and welcome, everyone. It's such a pleasure for me to introduce Dr. Christina Buchanan tonight. So Christina, you can pop up on the screen now. Great, thanks, Amy. So this is a little bit about Christina. So Christina has a long research history. She did a PhD in biological sciences at the University of Auckland and had quite a long lab-based research career. And then she wanted to work more closely with patients in the clinical environment. So she studied a Master of Genetic Counselling at the University of Sydney. So she's currently a study coordinator and a research genetic counsellor in the Neurology Department at Auckland City Hospital. So she's applying her skills in genetic counselling also to projects in neurogenetics. And she continues to hold an honorary academic position with the Maurice Wilkins Centre at the University of Auckland. So in 2021, Christina was awarded a Neurological Foundation project grant to study early onset Parkinson's disease in Māori and Pacific populations, and that's what she's going to talk to us about tonight. So thanks for joining us, Christina, and I'll hand over to you.
Kia ora. Thank you very much. I will share my slides and get started and do the formal introductions and thank yous. So tonight's webinar, the title actually came from the Neurological Foundation, the great minds at the Neurological Foundation, and it has inspired me to really put the Pacific at the centre of what we're going to be talking about tonight. But to begin with, I'm just going to do a sort of visual pepeha and introduce myself. So kia ora, Maori, talofa lava, malo alele, Bulavanaka, hello. So that sort of gives you an idea of the people I'm working with and also where I've come from. My parents, my father, Keith Buchanan, is from a little town called East Kilbride in Scotland. And my mother, Penny Buchanan, was born in Indonesia of Dutch and Indonesian parents. And even though these two people were born so far apart, they were both quite adventurous and attracted to warmer climates and found themselves in 1960 on a small little island in the Central Pacific, which is where I was born and where my siblings grew up. So mum and dad met and married on Barnaba, Ocean Island, in the Central Pacific there, and lived there for 13 years before eventually immigrating to Auckland. So Barnaba doesn't have much in the way of mountains, but my maunga would be Rangitoto. That's what I feel an affiliation to. That's the maunga I feel an affiliation to. And I guess the Waitemata and the Hauraki Gulf would be the water that I would also be drawn to, but that's all really part of the Great Pacific Ocean. And as you can see here, the Pacific spans all the way from Russia, China, and Southeast Asia, encompasses Papua New Guinea and the eastern coast of Australia. New Zealand is part of the Pacific, goes all the way over to the Americas in the east. So it's a huge expanse of water. And I guess we all take it quite for granted, but in the inspiration that I took from giving this talk with the title provided, it really sparked some further investigation for me of the things that I have taken for granted. So the Pacific is just massive. And this here shows us exactly how massive. The Pacific Ocean actually is able to encompass all the major land masses of the Earth in its watery mass there. So it's bigger than I think many of us realise. And within that space, there are thousands of islands. These have been sort of categorised and broken up into different areas, not by the people that live there themselves, but by cartographers and other people over the years. So there's Micronesia to the north, the small islands. There's Melanesia, which encompasses Papua New Guinea and expands out to Fiji. And Polynesia, which is a huge area, of which New Zealand is part. But it encompasses Tonga, Samoa, as far north as Hawaii, and as far east as Easter Island. And another thing that I thought I'd just find out for myself, because I've been listening to a lot of history podcasts, and there had been talk on there about, you know, the journeys of Christopher Columbus. When we put Christopher Columbus's journey, which was meant to be this great journey, into the Pacific, that 3,100 nautical miles would probably have taken him from the Philippines to about Nauru. So really, the Pacific compared to the Atlantic is just absolutely vast. And as one would expect with such a vast area and so many different islands, the people of the Pacific Ocean are incredibly diverse, and there's just so many different types. So spanning from west to east, I've included the maritime Southeast Asian islands, Brunei, East Timor, Malaysia, Philippines and Singapore. Going right through, there are so many different populations in Papua region, and here is only a sample of the Micronesian islands, and I had to add Barnabans to the list because that's where I come from, so that was quite important for me, but it wasn't even on the list. Melanesians, you will maybe recognise only a few different populations on that list. The Polynesians are also many and varied. There are two different parts to Polynesia, really. There's the Western Polynesian region and the Northeast and South Polynesian regions. So these sort of differentiations will become more relevant as the talk goes on. So I just, by introduction, just wanted to introduce the Pacific and its people and put them central to the story that I'm telling tonight. Now for the more formal part of the talk, going into a bit more of the sciencey stuff. By way of overview, I will be introducing Parkinson's disease and genetic Parkinson's disease. I'll be talking about the index cases that got us interested in this form of Parkinson's disease. The pilot study that we published in 2020 and 21, and which has led us to the studies we're doing now, and what we're learning about our people, the place around us, and the prevalence of this disease. So Parkinson's disease is the second most common neurodegenerative disorder in the world after Alzheimer's. In New Zealand, it affects about 1 in 500 people. So it's actually really quite common. The idiopathic form of Parkinson's disease, and I would almost call idiopathic like I don't know, because it can be caused by so many different things. Multiple factors, including just aging, injury if you've had brain injury. There could be genetic influences in there, and also the environment. So all of these things can combine together and form what we might be more familiar with, which is the later onset, sort of in the late 60s, 70s, 80s, the Parkinson's disease that develops like that. However, there are forms of Parkinson's disease, and a small percentage, about 10 to 15%, which is inherited. And many people might get worried by this and think, oh, is my form of Parkinson's inherited? The questions you need to ask yourself is there a really strong family history of Parkinson's disease? And also, is my type of Parkinson's disease and also the family history of Parkinson's disease, did it occur in younger people? And if you're saying yes to both of those questions, it is more likely that there may be an inherited cause, and so you might want to bring this up with your GP or your neurologist. But because Parkinson's disease is so common, it can be, you can still have a family history without it being in an inherited form of Parkinson's. But I'm focusing now more on the early onset form of the disease. So 10 to 20% of Parkinson's disease actually occurs in people relatively young. So we're talking early onset would be defined as under the age of 51. Worldwide, the most common recessive causes, so this is a genetic term, recessive means you have to have inherited 2 faulty copies. The most common recessive causes of early onset Parkinson's disease are due to changes in 2 genes. The first gene is called Parkin because it's involved in Parkinson's disease. And the second one has been called PINK1. So Parkin has been attributed to about 9% of early onset Parkinson's disease, and PINK1 attributed to about 4% of early onset Parkinson's disease. So if we break down those numbers a little bit more, because it's all these a percentage. If we had a population of 1 million people, we would predict a maximum of 2,000 people would have Parkinson's disease. Of those 2,000, 200 to 400 of them would have the early onset form of the disease. And of those with the early onset form of the disease, we might expect 18 to 36 with to have their disease caused by variants in Parkin, and maybe only 8 to 16 to have their disease caused by variants in PINK1. So PINK1 Parkinson's disease is rare. We're talking only 8 to 16 people per million population, generally worldwide. This is based on worldwide prevalence. And the other thing I'd point out from this cartoon down here is that this PINK1 Parkinson's disease, which only occurs quite rarely, can occur because of multiple different genetic changes in the PINK1 gene. So these are called pathogenic variants. And if you inherit 2 of these, they might be 2 different ones or 2 the same. If you inherit 2 of them, then you will develop a form of Parkinson's disease that comes on before the age of 51. So why does PINK1 or abnormalities in PINK1 cause early onset Parkinson's disease? And PINK1 and Parkin, that other gene that is implicated in early onset Parkinson's disease, actually work together to identify, label, and remove damaged mitochondria. So mitochondria are small little organelles existing within each and every one of our cells, or most of them. Well, actually all of them. And if you have problems with your mitochondria, they need to be removed and recycled. And what PINK1 and Parkin do is they work together to make this happen. So if you have damaged mitochondria, they will be labelled and removed as a result of PINK1 and Parkin working together. And you can see in this cartoon that I've made down the bottom. So if there's something that causes the mitochondria to be damaged in some way, in a person who has normal PINK1 and Parkin, they've got the right quality control machinery that kicks in, recognises that those damaged mitochondria, labels them, removes them, recycles them, allows for healthy mitochondria to be made, and the cell remains healthy. If you haven't got, good quality control machinery, for example, you've got genetic changes in those genes, the dysfunctional mitochondria, instead of removed, will just sort of stay there and accumulate and build up. And when the cell becomes clogged up with all these damaged mitochondria, it actually causes cell death. And so when that cell death happens in the dopamine producing cells, neurons in our brain, that's when we get the symptoms of Parkinson's disease. So I'm just going to introduce the two cases that got us interested in this a few years ago. The first person was a male. He presented at age 26, but when he was questioned further, it became obvious that he actually had some symptoms as young as 12. So he had developed a right arm tremor. He had stiffness and often frequent falls. When the neurologist saw him and prescribed dopamine, that dopamine helped him. So he was responsive to dopamine. He didn't report a family history of Parkinson's disease, and there was no report of cousins marrying cousins or anything like that. He was Samoan. At the same time, another man presented at the Auckland City Hospital Department of Neurology. This man was actually seen by my boss, Associate Professor Richard Roxburgh. So they were seen by two different neurologists. He had developed some symptoms around the age of 33, including right-hand slowness. He had some sleep disturbance and difficulty walking. He was also responsive to dopamine when he was prescribed it. He did report a family history of Parkinson's disease, but it was a few generations back on his father's side. And there was some talk of ancestors having Parkinson's disease, but not a lot of detail. He didn't report any intermarriage within the family, and he was Tongan. So these two people, one Samoan, one Tongan, but both with early onset Parkinson's disease. The two neurologists, Mark Simpson and Richard Roxburgh, both sent away for genetic testing due to the young age of these people that developed their symptoms. And both of them, unrelated, came back with being homozygous, which means they had two abnormal copies of the same abnormal PINK1 gene variant. And that's the scientific name of the variant, but basically all this is telling you is the name of the protein and the change in the protein, which says at position 3 to 47, there's a change from a leucine to a proline. So it's a single amino acid change, and that's caused by a change in the gene at position 1040 from a T to a C. So it's such a little thing, but it has big ramifications. That observation of two unrelated patients led us to undertake a pilot study. And Richard asked the question, if these two people, one Samoan, one Tongan, unrelated, both have the same apparently rare change in the PINK1 gene, how many other Pasifika or Māori patients with early onset or with Parkinson's disease might have this same variant? So his colleagues in the neurology department referred about 22 patients of either Pasifika or Māori ancestry. Of these, 8 were consented to look at the PINK1 gene, specifically looking at that single variant, change from the leucine to proline at position 347. 5 of the people, 3 Tongan and 2 Samoan people, had early onset Parkinson's. So they had the symptom at the age of 51. 3 of them, 1 Tongan, 1 Samoan, 1 Māori, had a later onset disease. There was a distribution of 5 females and 3 males in the 8 people we tested. And what we found was quite astounding. All 5 of the people who had early onset disease were positive, homozygous, so had 2 copies of this abnormal PINK1 gene. The other 3, the 3 later onset, still young, still, in their 50s, but they had a later onset disease, they were actually negative. They did not carry the same gene variant. This told us that what most people thought of as quite a rare cause of pink 1 Parkinson's disease, and was not so rare. And so it prompted us to investigate further, and we've applied, and that's when we applied for funding from the Neurological Foundation to do some further research. And our focus has always been on the people who have this form of Parkinson's disease and the people who support them, their families, because this is an inherited form of the disease. There is an extra connection and it's of great importance. And what we have found, and I guess it comes as no surprise, we've now, in addition to those 5 people, but including them, we've now tested 24 Samoan, Tongan and Tokelauan people, and 2 Māori. There are many less Māori that present with early onset Parkinson's disease. Of the Pasifika group, those 24 people we've tested, 19 of them have pink 1 gene variants that are causing their Parkinson's disease. None of the Māori patients, so there's only been 2 tested, but like I say, less of them in the first place, but none of them have tested positive for the pink 1 variant. So if we compare what this means for early onset Parkinson's in New Zealand, Pasifika Māori populations versus the rest of the world, that's what this infographic is. You might remember I said, so when you have early onset Parkinson's disease, I had that slide that said about 4% of it would be caused by all of these different variants in pink 1. So only 4% if you're in the world, if you've got early onset Parkinson's disease, a small percentage of you might be considering pink 1 as a cause of your disease. If you are Māori or Pasifika, or in this case, we would say Pasifika because we haven't found any Māori patients as yet, but this includes Māori and Pasifika, 73% of people have pink 1 as the cause of their disease. Thus 73% are homozygous for this one rare variant. We've found one person that has L347P and one other of the pathogenic variants, and about a quarter of people do not have changes in pink 1. So because we now have a cohort of so many people that have this disease, we were interested in finding out what has it been like for them living with an early onset form of the disease that it has affected them young, and living with Parkinson's disease when actually you might not be expecting to have Parkinson's disease. And indeed that's what my colleague, Dr Fuawhiwhi Fa'alau has found. So Dr Fa'alau has been running the Talanoa, so it's sort of a qualitative study where you sit down and you listen to people, you learn their stories, you learn what has impacted them the most. And she has found that there is no word for Parkinson's or dementia in Pacific languages. And the disease is often referred to as a pālangi illness and can carry a stigma. So I've heard this as well from the people that I've talked with. They are so surprised, the Pasifika people with Parkinson's disease, so surprised to find out they had Parkinson's because they thought that was a disease that only occurred in Pālangi or Pākehā people. And here's a quote from Fuawhiwhi. It isn't a disease that exists in a traditional Pacific worldview. Some other reason would have been given for the disease in the past, maybe a curse. And one of the things we're hoping through our studies is to demystify and uncurse this form of Parkinson's, and through that, the knowledge and understanding of what's causing it give people hope. So I'm going to come back to the Pacific again. I introduced it earlier, but we're going to come back to the Pacific and learn a bit more about what's going on and how this has got to New Zealand. So this map is the same one that I introduced earlier. And what we and other people have done, so at the same time as we were doing our study down here in New Zealand and we published our pilot study, we were looking at people who had published on the same variant, this exact same variant, pink one, L347P, and it had only ever been reported in 3 other places in the world, but all of those places were in the Pacific. So it's never been found in Europeans, in Chinese, in Latino. These are names given to populations by genetic databases. So it's not found anywhere else in the world, but it is found in the Pacific. And what these numbers here, these sort of fractions are, is what we would call carriers. So these are people that have one copy, one abnormal copy. The other copy is absolutely fine and is functioning normally. And so these people, because they only have one abnormal copy, they can continue on in life completely unaffected, as we're aware. However, they are at risk of passing that on. And if they partner up with another person who is also a carrier, then their children are at risk of inheriting 2 faulty copies. So Tan et al. In a control population who don't have Parkinson's, they found a carrier rate of 1 in 150. And Rogeva et al. Found in a control population of Filipino, they had a carrier rate of 1 in 33. So these are people that don't have Parkinson's. In Guam, in the Chamorros people, they had a carrier rate of 1 in 20. And we got together with our colleagues down at University of Otago, and we were able to access some DNA stored down there. And we found a carrier rate in Māori and Pasifika people of 1 in 34. So again, like I say, these are people who don't have Parkinson's disease. They are only carrying 1 faulty copy. Since we've started the study with the Neurological Foundation, we've also reached out to collaborators in Samoa and Hawaii to see whether what the carrier rates are like there. So in Samoa, the Olaga study has some DNA from control populations that don't have Parkinson's disease. They had quite a high carrier rate of 1 in 17. That means 1 out of every 17 people carries this abnormal pink one variant. Up in Hawaii, it was much less, 1 in 143 in Polynesian Hawaiians. When we separated out our numbers into what we call Western Polynesia, so this is all the Eastern and Western, so Māori and Pasifika. Western Polynesian we call come from this part of Polynesia, and the Eastern Polynesians come from this part of Polynesia out here. So Eastern Polynesians would include Hawaiian, Islanders, all through the Cooks and Marquesas, etc. If we separate out our control population, we see our carrier rate in Western Polynesians is around 1 in 18, which is actually very similar to what we find in the Samoans, whilst in the Eastern Polynesians or the New Zealand Māori, we have a report of carrier rate of 1 in 137, which is quite similar to the Hawaiian Polynesians. So this is telling us that this variant is throughout the Pacific, and it's travelled at a high carrier rate through to Western Polynesia somehow. We don't have any information through here, but on the subsequent migration that happened later to Eastern Polynesia, it doesn't seem to have carried on at the same carrier rate. When we look at databases, so these are the databases that geneticists might look at, and this one's called NOMAD, and these are how the populations are listed. So here we've got, these are control populations, they don't have Parkinson's disease. We've got 3 people out of, this is an allele number, so divide this number by 2, so we've got about 9,200 people here. People out of 9,200, and they're East Asian. And actually, when you look into it more, they're Filipino. And down here in other, so 1 person out of 3,000, but other includes Pasifika. So that this could be Pasifika people, but this particular variant is not found in African, Ashkenazi, Jewish, Europeans, whether they be Finnish or non-Finnish, Latino or South Asians. So those carrier rates are really quite astounding. And what does that mean to the prevalence of Parkinson's disease in New Zealand? We have some information about the prevalence in different populations in New Zealand. On the whole, it's higher in European populations. So this is the number of people who have Parkinson's disease per 100,000. And this is the study it comes from. So Europeans do have a higher prevalence than other populations. And I've just listed these from high to low. And also because there are more Europeans in New Zealand than the other populations. I think I've got the wrong number there, sorry, but anyway. Because of that, there are many more people living with Parkinson's disease of European descent in New Zealand. And I guess that's why most people would think that Parkinson's is a white person's disease. And from our numbers of, based on international studies, we might predict as many as 58 of those Europeans might have Parkinson's as a result of those multiple different variants in pink one. But really the striking thing is this number down here in the Pasifika. So there's a lower prevalence, a lower population, so a lower number of people living with Parkinson's, but of those numbers, it could be the absolute maximum. This is an absolute maximum because we're talking about, we only know about certain parts of the population, and we're relying on people marrying within the population. So absolute maximum could be as many as 294. Based on the carrier rate in Māori is much less. So when we're talking to people about the fact that they have this early onset form of Parkinson's disease and that it's genetic, they obviously they will jump to being worried not only for themselves but for their family. And usually, because of recessive disorder, usually you're only at risk of passing on one copy. Well, you are, as the affected person, you will only pass on one copy. And usually you'll be partnered up with someone who doesn't have anything wrong with their PINK1 gene. So all the children you would expect to be carriers and not affected by Parkinson's disease. So this would be very good news. However, in this population, because of that high carrier rate, there is a 1 in 18 risk that the partner might carry the PINK1 gene, or the abnormal PINK1 gene. And if that were the case, it would mean that every child that this partnership had would be at 50-50 risk of either having a carrier or a child affected who would eventually grow up to have early onset Parkinson's disease. So I think you can see here that the high prevalence of this PINK1, early onset Parkinson's disease in New Zealand, means that it has a high negative impact in our population. And that's why I feel that prioritising research into PINK1 Parkinson's disease has a potential for high positive impact. In addition, we can say that focusing on PINK1 and mitochondrial function in Parkinson's disease will also have a positive impact for the more common idiopathic forms of the disease. So the key points I'd like you to take away. We know that the PINK1 L347 abnormal gene change has been observed in New Zealand and other Pacific countries, and that it is carried at a higher rate in some Pasifika populations. I'd like to emphasise the fact that since we know what causes it, we know we have a target to focus therapies on, and that a cure for PINK1 PD would have huge implications for Parkinson's disease prevalence in New Zealand and the Pacific, and a positive impact for other forms of Parkinson's disease. So by way of acknowledgement, I really want to, again, first and foremost, thank the patients and participants who have taken part in this study, whether it be clinically or as part of research. I've made great connections within the Pasifika Parkinson's community, and that's been forged by Aloisi Lafono, who runs the Pasifika Parkinson's Support Group in South Auckland. Dr. Fuafifa Fa'alau and Miraneta Lamalu, who research assistant, have been doing wonderful things working with people. We're actually doing the Talanoa in Samoan and English and translating it as we go. So if people are more comfortable talking in their mother tongue, then that can be accommodated. The ongoing neurologists and clinical staff are listed there that are taking part in our ongoing studies. And the previous pilot study and population study work lists yet more people, and you can see how many people have been involved in this study today. And it's been funded by the Neurological Foundation of New Zealand and the University of Auckland through the PBRF. But I would like to just leave the last words to a person that I rang up to invite them to take part in our research. And this was a quote, and I keep it there, I have it in my mind. And they shared, when I said my name and said why I was calling again to talk to them, they said, "Oh, I was really hoping you were ringing to tell me you had a cure for my Parkinson's." And whilst we don't have a cure yet, this we do feel is one of the types of Parkinson's because we know the cause. We are working hard, gathering this information and working towards a therapy, and we hope that it will be one of the first forms of Parkinson's disease to be cured. Thank you for your attention and joining me tonight.
Well, that's amazing, Christina, just really fascinating. And I'm sure we're going to have lots of questions coming through now. I was hoping that I might be able to start with one of my own, just as you were talking about it being such a rare disorder, but researching it could have a really high impact. What is the likelihood of being able to get the type of funding you need to continue with this research when it is such a rare disorder?
So that's, so worldwide, it's a rare disorder. Obviously in New Zealand, it's less rare. Through the work that I've been doing recently, I've managed to hook up with a lab over at University of Dundee, which is run by a neurologist researcher called Maratul Murkit. And Maratul has been focusing for the last decade or more on PINK1 Parkinson's disease. And we are literally, I feel, right on the cusp. So there's a first paper published by a company with a therapeutic target for PINK1 Parkinson's disease called mitokinin, and that was just published in 2023. So it's all coming together at the right time. So in terms of targeting international funding, I think it's harder, but I think with the way funding is focused in New Zealand on Māori and Pasifika, I think there could be some avenues that we can explore there that can really help these people, and that will have the knock-on effect for other people.
And because you're characterising it in the New Zealand population, then New Zealand should be able to get access to this exciting new treatment that could be coming.
Yeah. Look, it's all very early stage. It's still at the discovery stage, and I've been working in research for long enough to know that these things take time, and it's about being realistic with how things progress. And but yeah, since we have an identified cohort, we have more people in New Zealand with PINK1 Parkinson's disease likely than anywhere else in the world, other than, say, the Pacific Islands. So we will be the best people to work with and partner up with if it should move on to clinical trials.
Well, that's fantastic news for the patients, and I hope that that continues to move along at a good speed. So with it being quite rare, and obviously the idiopathic version is more common in New Zealand, so how do you identify if you've got a strong family history in Parkinson's disease?
Yeah, so this is one of the, really what you when you come along, if you have Parkinson's and you're interested in getting tested to see if you have a genetic form of Parkinson's, look, the best way to get tested is to talk to your neurologist. Your neurologist would hopefully take a family history, and based on that family history, they would determine whether it's worth getting tested. A strong family history, so there are genes that do cause what we call autosomal dominant Parkinson's disease, so LRRK2 and others. If you had autosomal dominant, you would expect that maybe one or other of your parents might have had Parkinson's disease, and maybe if it's on your father's side, you might expect others on your father's side. If it's on your mother's side, you might expect others on your mother's side. So having multiple dotted around on different sides of the family doesn't actually build a stronger case. We're looking, a geneticist or a neurogeneticist would look for trends and work out from that family history whether it is likely or not. Genetic testing is getting cheaper. So one of the things that used to stop people getting tested in the past was the cost, because it's all publicly funded. But if as costs reduce, this is one of the things that we can do. I guess the thing is, it won't necessarily change your treatment, because you're still going to be treated in the same way as anyone else with Parkinson's disease. But what it will do is it will inform you as to what the risk is for the rest of your family.
So in your research, have you come up with any theories about why the PINK1 variant is so prevalent in Pacific populations?
So I'm not an anthropologist or I'm a biochemist and a geneticist. What we do know is we know that migration came from the West, from Southeast Asia into the Central Pacific and Polynesia. And we know that that variant was in Southeast Asia. So you would assume it's migrated into the Pacific. What is interesting is that that there is this complete change when we look at the Eastern Polynesian migration. So the migration up into Hawaii and down into New Zealand, which happened about 800 years ago. So they have a lower carrier rate. Now, carriers are just as strong as anyone else. And it's just one of those, we don't know as yet, but it's one of the things that we want to study. We hope to put all the DNA of the unrelated patients from Malaysia, Guam and New Zealand onto a chip and see how distantly they're related. And that will give us some information moving forwards about how it's come into the Pacific.
Do you think there could be an advantage to having a defective copy of the gene if it's survived so well on the move across the Pacific?
Again, when you've only got a single defective copy, it doesn't appear to impact you. It's only when you have the two defective copies.
So most of the time it's probably moved undetected across, yeah. So we talked a little bit about the potential that you've characterised these patients so that they can hopefully, you know, be a great cohort of people for any future therapies that come up. But can you elaborate on any other ways that your work has been impacting these families and their clinical care?
Yeah, so by identifying, at least I can comment on what's happening at Auckland City Hospital, which, because Auckland is the centre for, or has the largest population of Pasifika people living in New Zealand, that's a great place to start. So what we have is a neurologist who is a movement disorder specialist. His name's Dr Vish Dayal, and Vish is going to become the Pink One specialist. So neurologists all have their own different specialties, whether it be stroke or epilepsy or whatever. And so part of the problem we've had in the past is that these people are seen by multiple different neurologists, and the 2 and 2 hasn't been put together until quite recently when we found this genetic link. And so by having a single neurologist who is focusing on these people and can really provide that wraparound clinical care, that will benefit them hugely. And also by reaching out, and I've linked them up with free physiotherapy provided through the Duncan Foundation. There's Rope Neurophysiology neurophysiotherapy is providing free physiotherapy for people with early-onset Parkinson's disease, Pasifika people with early-onset Māori and Pasifika. And yeah, just involving them with that Pasifika Parkinson's support group that LOSE runs. There's huge benefit from being part of a community and not feeling alienated and like you're the only person with this disease.
That's really fantastic to hear about this community of clinicians and other therapists that you're bringing together to help these families. I can't imagine what it's like to be diagnosed with Parkinson's at the age of 26. It must be a huge shock.
Yeah, absolutely.
So new technologies like CRISPR, this comes from one of our audience who's obviously got some scientific knowledge. Are some of these technologies helping with your work?
Yeah, CRISPR's a bit further down. There are definitely genetic therapies that are starting to happen. They usually work at the mRNA level rather than the DNA level just at the moment. And the benefit of Pink One is that we can target it at a biochemical level. So we know what's happening to the kinases, we know what's happening in the mitochondria. So we have a few different ways, or scientists have a few different ways of attacking the problem, whether that be through small molecule therapies that can upregulate the PINK1 pathway, or whether it be through genetically rectifying the changes in the cells in the body. So I guess with CRISPR, you're wanting to, it is trickier because you're wanting to change every cell in the body, whereas CRISPR technology seems to be better when it's targeted to a particular organ just at the moment. But who knows, these things are rapidly changing.
Oh, thanks for explaining that. I know you talked a little bit about how Parkinson's disease was not widely thought to be in the Pacific at all. So getting people together and talking about their families and their experiences, is there any awareness in those communities of this disease?
Yeah, so there's a growing awareness. Like I say, I've reached out to the Pasifika Parkinson's support group, and I've done talks through that support group. They are linked up and sort of supported themselves by Parkinson's New Zealand, which is a nationwide charity that supports people with Parkinson's. And so what I do is, if I find out that someone wants to be part of that Pasifika Parkinson's support group, because I've been made aware of them through neurologists or the movement disorder specialists or someone like that, then I will link them up with Parkinson's New Zealand, and so they get the wraparound service that PNZ can provide, as well as linking them up with the specific support group that can really make them feel like they're not alone. So yeah, there's multiple ways of linking people up, but in the first instance, it does rely on them reaching out to get a diagnosis and then feeling like they want to get further help. One of the initial periods that people go through is they will hide themselves away. They'll be like in denial of their disorder, and they'll think like they're the only person around with it. But once they move through that and can reach out, then there's huge positive impact for them. And I'm working together with other researchers in this field to really try and minimise that denial period and give people the linkages as soon as possible to reach out, whether it be to support groups or research or physio, all the things we know that can really help people in those early stages.
So that leads quite nicely into a very good question from the audience. If I know I have PINK1 Parkinson's, how can I be part of the study?
So the easiest thing would be to Google my name, Christina Buchanan, PINK1 Parkinson's, and hopefully it would come up with an email address for me. But also Neurological Foundation would have my email address, and I'm happy if anyone is ready to engage, then I'm happy to talk with them, explain the different types of research that we have underway and how people can get involved straight away, and also what we've got planned for the future.
Well, thank you for very generously sharing your email. I saw that it was on the beginning of the slides as well. So when this talk goes online, your email address will be on there that people can access as well. So that's very generous of you. Thank you. So we're now at 8 p.m. Would you like to do a couple more questions before we close?
Yeah, I'm happy to. Yeah.
So if other people around the world with early onset Parkinson's don't have the PINK1 variant, what is causing their disease? Is it the Parkin variant?
Yeah, so like I said, 10 to 20% of Parkinson's disease occurs early. Of that 10 to 20%, some proportion of it will be caused by genetics alone. some other proportion of it will be caused by multiple different things. So there are 6 genes. There's a panel of 6 genes that you can get tested for, and that will test the 6 most common genes that cause what we call monogenic, single genes that cause Parkinson's disease. So PINK1 we know is important in the Pasifika population, but if you're not Pasifika, we would, and you have quite an early onset form of the disease, we would probably send you away for panel testing and test all 6 of those genes, because we wouldn't just assume that it's going to be PINK1.
So is the defective mitochondria and the problems with them being cleared, is that only associated with the PINK1 and Parkin variants, or is that something that happens in all cases of Parkinson's?
So mitochondria, because they are the powerhouses of our cells, we know definitely PINK1, Parkin and DJ1, those 3 genes cause problems with mitochondrial function and clearing mitochondria. There are other forms of the disease that result in mitochondrial dysfunction and have the knock-on effect, but it's not a genetic problem. It might be some sort of neuroinflammatory response that causes mitochondrial dysfunction. And so there are other ways that mitochondria might be involved in cell dysfunction. So it's not purely genetic, but the mitochondria we know are essential for happy cells and for functional cells. So that's why the mitochondria themselves are a focus. Even for researchers overseas who aren't focusing on PINK1, there's plenty of research going on into mitochondria and how we can boost their function.
Do you know of any other research going on in our part of the world about why the PINK1 gene is so prevalent?
No, there's an Australian group that just published recently their finding also that obviously in Pasifika people in Australia who have early onset Parkinson's disease and lower limb dystonia or dopamine responsive dystonia. So that's still at a clinical level. They're neurologists. I'm not aware that they are starting to do actually any in-depth research in the area. But obviously there are big Pasifika populations in Australia, so this will be impacting them too.
And can we learn about the general Parkinson's disease or the idiopathic Parkinson's disease from studying the PINK1 gene? Are there things that will help uncover?
Yeah, because we know there's a series of pathogenic variants in PINK1. There are also other variants in PINK1 that don't cause monogenic forms of Parkinson's disease, but who knows, maybe a change somewhere else in the gene together with an environmental change, some brain damage from boxing or something like this. These things together could be multifactorial, and there could be multiple different changes, genetic changes together with. And I see Richard has raised his hand and he probably has a comment to make. I don't know if we can turn him on. You might have to put your comment in the chat box, Richard.
Yeah, send us your comment, Richard, who is the neurologist at Auckland Hospital who runs the neurogenetics clinic and works very closely with Christina. So we'd like to hear if Richard has any further comments to add. But while we're waiting to see if he pops something in the chat, I'm just going to close with one last question, which I think was a really good one from our audience. What are the benefits of doing this research in Aotearoa, New Zealand?
I think the benefits are that So there's multiple different benefits, whether you've just got your research hat on or whether you're a person living with Type 1 Parkinson's disease. Obviously, if you're a person living with Type 1 Parkinson's disease, doing research in this area is your hope for the future. If you are a researcher and you're thinking about the benefits of this research, it's the one place in the world where we should have a drive. This is impacting our people, and it has we can make the biggest change for a large number of people by doing research in this area, whereas overseas it's a far rarer disease and therefore has less impact.
That's fantastic. Well, I can't see any comments from Richard in the chat, but Amy can let me know if I've missed something. But I just want to say thank you, Christina, for sharing all this fascinating information with us tonight, and also just seeing the group of people that you've brought together, the clinicians, the families, people who can speak with them in their own languages and do the qualitative research, as well as therapists to work directly on their physical symptoms. And just that slide that you put up at the end, you know, all those names on it of all the people that you're working with, and I recognise so many of those names, and just a really great team that you've built around you. So yeah, well done for the research that you're building, and thank you for taking the time to share it with us tonight.
Thank you. I really appreciate the support that the Neurological Foundation has given this project as, you know, a kickstart, and hopefully it can be the basis for some really great impactful change.
Well, I'll say a big thank you to the audience as well. Thank you for joining us tonight. We hope you all have a fantastic rest of your evening, and Amy will be closing the webinar very abruptly very soon. Good night.
Thank you.